Yeah, my name is Nicki Watson, and we're going to be talking today about sample prep for biological imaging for electron microscopy. I've included my email address there, so if this talk inspires you to think about doing some EM biological, we can exchange some emails so we can get ready and get going once we return back to normal. I'd like to start with this slide here, just as a reminder that biological imaging and microscopy imaging has been a cornerstone of biology from the very start, from the very first Leeuwenhoek microscope to then the more advanced, sophisticated microscopes, until we get into the Zeiss microscopes of the late 1800s. If we fast forward about 400 to about 50 years from the beginning, we see our first electron micrograph of an intact cell. This was a chicken fibroblast, which lends itself nicely to this sort of work because it can grow very, very thinly so that we can get the electron beamed through it. This picture actually represents the beginning of the field of cell biology. It was created with Keith Porter and Ernst Fulham in 1945 with the group at Rockefeller Institute that was working to create electron micrographs for cell biology. Ernst Fulham was a chemist working with the microscope companies, and electron microscopes were very new at the time, probably not more than 10 years old as a commercial instrument. And they were very interested in working with biologists to see if they could use this new instrument as a way of seeing more deeply into cells, seeing high resolution. And so the Keith Porter group was collaborating with them on this goal. Then we come to George Palade. He was also part of this Rockefeller group. In 1974, he was awarded the Nobel Prize of Physiology and Medicine for developing methods of studying cell biology with an electron microscope. He did this work in 1955, his first notable discoveries being the characterization of ribosomes and endoplasmic reticulum. And because of this work, he's often described as the most influential cell biologist ever. Basically, before this work, there was no cell biology to speak of. So biological electron microscopy was the state of the art for cell biologists through the 1990s and through 1999 until Roger Chen developed green fluorescent proteins, and confocal microscopes became readily available to research labs and were relatively easy to use. And so confocal microscopy took center stage for about 20 years. But now we're seeing a rebirth of biological electron microscopy. This is due to two primary factors. One, the Human Brain Project is getting a lot of people interested in finding and developing methods for being able to image the entire human brain at the resolution of an electron microscope. But also the high resolution cryo TEM revolution has also gotten electron microscopy back into the headlines. In 2014, Birgit Karager published the first cryo electron micrograph of a structural image with a resolution of finer than 3 angstroms. So this was a milestone that had been trying to be achieved for a long time. And then in 2017, Dubochet, Frank, and Henderson were awarded the Nobel Prize for their work in developing cryo-electron microscopy methods. So methods and sample preparation for biological electron microscopy has always been difficult, and when those milestones are achieved, they're major breakthroughs. So what this tells us is that biological electron microscopy requires special and careful preparation of our samples. The goal here is to look at biological tissue, and we want it to look as it did when it was alive, but when it's in a microscope under high vacuum and being bombarded with electrons. This is the challenge that we had to overcome. Also, it's good to keep in mind that not all preparations are the same. The preparation that you are choosing is going to be specific to the material under analysis and the desired information from that specimen. So, before you get started, you want to clearly define what your question is, and then the sample preparation method will depend on that question that you're asking. There are three primary questions that you can be looking for answers to when you're doing cell biology or biological TEM. First, we would be interested in the morphology of our material, the ultrastructure, the membrane concentrations, the shape, size, and organization of organelles. We also may be interested in structural information of purified proteins, purified organelles, things along this line. And lastly, we can look at protein localization at a high resolution. And we would do that with an immunolabeling preparation. So let's talk about structural data first. When you are preparing some sort of protein, either completely in a test tube or if you're purifying it from cells, when you've got this protein purified, you're going to want to take a look at it and see what it is, what it looks like, and how it's organized. So in this example, we have amyloid fibers that are relatively easy to make, but things can go wrong and things can become aggregated, things can be contaminated. So we want to get a quick look at these things by EM first. We would use negative staining to look at these samples. We're actually putting our sample in negative contrast against a dark background of heavy metal stain. The sorts of structures or the sorts of samples that lend themselves to this perfectly would be if you're making nanoparticles, negative staining is a great way to get a quick look and QC the quality of the nanoparticles you're preparing, looking at the size, shape, density, concentration, cleanliness as well. Some other examples of nanoparticles on this side. If you were to take a reading of this from an optical density instrument, you might get a different answer than what you're going to get looking at the images directly. So you're QCing nanoparticles again in this way. You can also look at the purity of purified organelles. In this case, there's a bunch of vesicles that have been purified from a cell prep, and we can look at that very quickly and easily to see the quality, the concentration, cleanliness of the sample. We can also look at small whole cells. In this case, we have a whole bacteria cell. What we were looking for here was whether or not the cell is actually expelling these tiny micro vesicles. You can see a flagella on this end of the cell. What we can't know for sure by negative staining is whether this ruffles and convolutions on the surface of the cell are real. So this is one of the downsides of negative staining, because the sample is dried. But in general, negative staining is going to be a fast, easy, qualitative method for examining structures of isolated molecules, small cells, or purified organelles. An electron transparent sample is surrounded by the electron dense stain, and the specimen appears in negative contrast. So how do we do this? First, we need to prepare our grid. A grid, if you're not familiar, is usually copper, but you can use nickel or gold, mesh grid of the mesh size between 200 and 400 microns. We coat this grid with carbon or a formvar covered by carbon substrate, because we're going to be looking through the holes in the grid at our sample. We want to freshly ionized glow discharge, is sometimes called, these grids so that you allow for more evenly deposited material in a more uniform stain. The protocol is quite simple and easy to master. The most difficult step is, before you ever prepare your sample for electron microscopy, is preparing your sample from the cells that you're working with. After you have a clean sample prepared, you simply absorb that onto the grid, or you can float the grid upside down on a 10 microliter drop of the sample for 30 seconds to a minute. You wash this, pick this up with fine forceps gribs, wash it with 5 drops of your uranyl acetate, draw off the excess UA, and let the sample air dry. Now what we have achieved again is your material is actually embedded in the stain. Now there are as many protocols for negative staining as there really are people doing negative staining. But this is a protocol that I've been working with for many years, and it's pretty fail-safe. The things you might change would be drying your sample on the grid before staining, but that's really the only way that you might change the appearance of your sample. The most common stains that we're going to be using is 1 to 2% aqueous uranyl acetate. Phosphotungsten acid is often used, and ammonium mobilate is often used. These heavy metal stains have different molecular sizes, and so may give you different resolution levels of fine structure. And they also are notably used at much different pH ranges. And it seems to be the pH of your stain and the pH of your protein can interact with each other to produce better or worse results. This is something that you just typically try to work out as you're going along. I would always start with 1% aqueous UA. That's going to work with 95% of anything that you're trying to stain. The challenges that we're going to run into with negative staining: first, your sample has to be a monodispersal of particles. You're not looking at overlapping structures that are going to interfere with your resolution. Compatible buffers are important, and that means compatible buffers with the stain, but also with your protein of question. You want to have a buffer that keeps your protein in a native state, but also doesn't precipitate out all of the UA. If you can get away with water, that would be even better. Structural artifacts are common, and you need to be able to recognize them. Mainly flattening of spherical structures is a common artifact that people can misinterpret. You can also have precipitated UA that may be blocking the resolution of your structure as well. The other challenge is your sample is air dried, and biological samples tend to want to be in a hydrated environment to maintain their native structures. So air drying is going to affect what they look like finally. Sample concentration is critical, as well as the cleanliness of your sample. If you've purified vesicles or fibers and you haven't removed all of the other extracellular material from your sample, it's going to interfere with what it is you're trying to image. Finally, the stain itself is going to interfere with the resolution of fine structure. So while this is a great way of getting quick and easy images of your protein, the resolution is going to be limited to about 1 nanometer. The benefits, however, is that this is a really high contrast, fast, easy sample prep that is very easy to master. Negative staining is also the first critical step if you're planning on going to high resolution cryo electron microscopy. In this picture, we have a perfect sample for cryo EM. It's clean. It's the only thing present is the protein that we're looking for. And we're lucky in this case that we have almost an infinite array of orientations of this protein. So by using computer generation, we can take all those infinite arrays and then start doing our 3D reconstruction and start averaging images to get better resolution of this image. So for higher resolution, angstrom level resolution, we would need to go on to the cryo-EM level. I'm not going to talk about that today. I'll leave that to Svetla McPhie. She's giving the master's class on this on June 3rd. But the basic take home why we get higher resolution from cryo-EM is because we're looking at hydrated, unstained biological materials that can be imaged directly. So the next questions that we might consider ourselves with biologicals electron microscopy is the ultrastructure. We want to look inside the cell, we want to see cross sections of the cell and see what that looks like. This is referred to as routine morphology or classical electron microscopy. We're looking at ultra thin sections of plastic embedded material, and the sample prep here is more involved. We have to first fix, dehydrate our sample, embed it, then section it, stain it, and then finally we can get to the microscope. The sorts of data that we can get, though, can be very powerful. Here we have a toxoplasma infected cell. Toxoplasma is a parasite that is very dangerous to pregnant women and elderly and immunocompromised people. So we wanted to see how this parasite can infiltrate the cells, what it does to the cells when it gets in there, how it's organized, how it reproduces and these sorts of things. But we can also see what we gain by classical electron microscopy sample preparation techniques. If we look at the cell here, we can see the nucleus very clearly. We can see the nuclear envelope. We can see nuclear pores in cross section. Here we have a nuclear pore that still has a bit of a fenestration. We have the Golgi apparatus here, nicely organized stack, rough ER, some dense, electron dense particles. So we can learn a lot about the ultrastructural morphology of this parasite by doing this sort of classical EM. We also have an opportunity to look at important model species. We have our bacteria here. Now when we've prepared the sample this way, we see that this bacteria does not have a ruffled, convoluted surface, but it is smooth. We can see the internal organelles. Bacteria don't have a lot of internal organelles, but we can see them clearly outlined around each other. So you see them as rods, see them in cross-section. Yeast are another important model species. Geneticists use yeast and mutate their genome, and if we can get high resolution ultra thin sections of the yeast, then we can start examining how excuse me, how we can examine how we've manipulated the ER, for example, or the mitochondria. And again, this plastic embedding is the only method that we would have to be able to see the internal structures of these cells. So the sample preparation methodology is more involved than with negative staining. We start with chemical fixation. If we're looking at cultured cells, then we would want to, after fixation, scrape the cells off the plate, spin them into a pellet, stain with osmium tetroxide and uranyl acetate, fully dehydrate the sample, infiltrate with our plastic, put that into a molded block and polymerize the plastic at 60 degrees. So we have a very hard block. A hard block allows us to cut very thin sections. So that we would section, pick up those sections, stain them again, because even though we've infiltrated with heavy metals, once we've stained these sections, They are still essentially transparent, so we must stain them again. And then finally go into the microscope. This is going to be approximately a 2-week preparation time where you would do your embedding the first week, your sectioning, staining, and scoping the following week. So to go into this a little bit more detailed about why we're doing this, what's important, and how we've developed the methodologies that we have. With fixation, we want biological material changes very rapidly when it is not in its warm, safe, humid environment. So cultured cells can change their ultrastructure very, very quickly when you take them out of the incubator. So your fixation needs to be extremely rapid. And that's not just rapid of putting the fix on, but the chemical reaction of the fixatives infiltrating the cells. Mitochondria, for example, are very, very susceptible to any sorts of environmental changes. So if you're studying the appearance, morphology, or interactions of mitochondria, say with ER, then fixation needs to be addressed very, very carefully. So again, our goal is that this preservation of the structure looks as it did when it was in the living state. We want no fixation artifacts, and that happens when you fix rapidly. The way we also are protecting the cells from disruption during the dehydration phase. The fixation that we're using is called Karnofsky's fixative. It is the gold standard, and it is especially the gold standard when it is made fresh from granulated EM-grade paraformaldehyde. If this is not available to you, you can buy Karnofsky's fixative from Electron Microscopy Sciences. Karnofsky's is made up of 3% paraformaldehyde, which is a small molecule that penetrates quickly and starts the fixation process. 2% glutaraldehyde is a larger molecule, but provides the cross-linking between proteins that will create a permanent fixation of the cells. We have 5% sucrose and 0.1 molar sodium cacodylate buffer for osmotic balance within the cell. We don't want to have our cells shrinking or swelling as a result of our fixation. So we fix our cells for an hour, room temperature. Then we wash them thoroughly with sodium cacodylate buffer, and then stain with our secondary fixative, osmium tetroxide. Osmium is going to react with lipids, membranes, and nucleic material and create an electron-dense contrast in our final product. We're going to fix with osmium for about an hour, and you want to do that on ice so that you don't precipitate osmium throughout your cell. So it's also important that you trim your samples to not greater than 1 millimeter square, because osmium and the glutaraldehyde is not going to penetrate any deeper than that within your sample. So you want to trim them small so that you get good rapid penetration of your stains through to the center of your sample. We follow that up with uranyl acetate, which is a heavy metal, binds to the osmium, and again gives contrast to the membranes. Typically, I use uranyl acetate overnight and begin my dehydrations the next day. So dehydration, why are we dehydrating? Biological cells are essentially water balloons. We need to remove this water so that we can go into the vacuum of the microscope, but also so that we can penetrate it with the hydrophobic embedding resins. We commonly use EM-grade ethanol, and we start slowly with 50% ethanol to 75, 90, 95% ethanol, and then 3 changes of 100% ethanol. You want to make sure this is absolute 200 proof EM-grade ethanol that you're using. Then we're going to transition slowly into plastic by using propylene oxide, and we're going to start, again, transitionary fluids. We're going to do 50-50 propylene oxide, 100% ethanol, 100% propylene oxide, 50-50 propylene oxide plastic. I will typically put this overnight so we have a slow and complete infiltration of the plastic. The last day, we're going to have multiple exchanges of 100% plastic on a rotating, or on a rotator so that you have good sample contact with the plastic. Sample is then placed in a proper orientation in a mold, and you want to give some thought before you finally embed your sample. You want to have in mind what sort of orientation you want your sections to be. For example, if you're looking at muscle tissue, do you want cross-section cuts of the muscle or do you want longitudinal cuts of the muscle? So that once you embed this and polymerize the plastic, you can't undo this. So you want to give some thoughts to how you want your final picture to look before you can finish a step that can't be undone. All right, so then we're going to bake 24 hours, 60 degrees C in a dry, specifically designated oven. Water is our enemy here. We don't want any water around when we're in the 100% plastic. The standard epoxy embedding resin is Embed 812, formulated as the hard recipe. There are other embedding resins, LR White, and can be used with a little bit of water, so you might be able to do some immunolabeling with that. Spurs is another resin that can be used. It has a lower viscosity level and is often used when we're embedding bacteria, plant material, yeast, or other samples or tissue or cells that are difficult to embed. The concerns we have here is that propylene oxide and 100% ethanol evaporate very quickly, so you want to be very careful that your tissue does not dry out when you're changing solvents. You want to completely dehydrate your sample, so if you need to do it more slowly because you're dealing with plant material or bacteria, do it more slowly. You don't want your solvents to absorb water, so when you get to your 100% absolute alcohol, use a fresh bottle. Old bottles may have dissolved water in them. Very important, fixatives, osmium, and resins are toxic and carcinogenic, so you want to work in a proper chemical fume hood. And you also want to be careful about the material that you're processing your samples in. Plastics and propylene oxide can dissolve some plastics. So you want to make sure that, say, if you're flat embedding cells in a dish, that your dish is not going to dissolve before you get it embedded. All right, so now we've got our sample embedded. We have a nice hard block. It's time to move on to the next step, which is sectioning. So sectioning involves trimming the block, facing the block, and then cutting sections. So the top picture, we can see our black piece of tissue here within this plastic block. And we've trimmed away all of the plastic that doesn't contain tissue. And we can rough face that. Once we've rough faced that, then we can take the block and do a final crisp trim on it. We want this base here to be no longer than 0.5 micron. Then once we've got it on the microtome, we can cut a section, and this is what we would end up with. So what are we going to section with? We have glass knives or we have diamond knives. Glass knives you'll make yourself with a glass knife maker. It takes a bit of practice, but can also be easily mastered. We start with a full-length piece of glass that is specifically designed length and thickness. We're going to break that glass in half and in half and in half until we end up with a perfect square. We're going to then break that perfect square on the diagonal so that we have hopefully 2 45-degree knives. While that's not likely to happen, what we generally end up with is knives that have a knife on one edge and a flat edge on the other. This means that this break did not happen at the 45 degree, but slightly off. We call this edge the knife, we call this edge the foot, and we're going to judge the quality and sharpness of this knife by the size of this foot. So if you're making multiple knives for different purposes, you want to choose the best ones for your final cuts, and you would judge the best knife based on the quality of the foot or its opposing side. If we turn this knife up, so we're looking at the cutting edge, we'll see this stress line, which is the fracture line. And this will also tell us the best parts of the knife to use. On this edge, the thickest part of the stress line is our dullest part of the knife, but it's the most durable part of the knife. On the shortest end of the fracture line, we have the sharpest part of the knife, but the most fragile part of the knife. So for general purpose use, we generally stick to this middle third of the knife, which is going to be the best of all worlds. However, you can, if you need to, just move from one side to the other. So once we've got ourselves capable of cutting ultra-thin sections with a glass knife, we can move on to using a diamond knife. A diamond knife is in many ways easier to use because it's a perfectly smooth surface. It's manufactured to have a hydrophilic diamond surface to draw water up to its edge. It's jewel quality diamond crystals. You can buy them in sizes from 1 to 6 millimeter. Typically people use a 3 millimeter knife. Maximum sectioning stability is assured because the diamond is welded into a stainless steel shank and then mounted onto the boat that you can purchase these boats in different sizes for different applications. So now we have our knife, we have our trimmed block. So we can start sectioning. This is also called ultra microtomy. So our polymerized block is trimmed to a 5 millimeter square or trapezoid with a very sharp razor blade. Using a glass knife, we're going to face this block until we reach our material or until we reach a mirror smooth finish. For beginners, you can then mount a boat onto a knife, fill that boat with water, and collect your sections. If you are at the skill level that you're using a diamond, you'll retrim the block, change to the diamond knife onto the microtome. Carefully align your block to the diamond. Slowly approach the block with the diamond and start cutting. We do need to be careful about the water level in the boat. We want this to be just the perfect level so that the samples come off the knife and float on the water. We don't want the water too high of a level because that would wet the face of our block and prevent cutting. We don't want it too low because then the knife itself would be dry and could get damaged and also can start damaging your block face. So now we can set the microtome to automatic cutting mode. We want to set our section thickness. We want to set the speed of the cut and the return speed of the block to the cutting surface. We're going to be cutting sections. Our goal is 50 nanometers thick for ultra resolution of ultra thin sections. We're going to pick up these sections on a grid, stain them, and examine them. So the thing you need to be concerned about when you're getting to using a diamond knife is that diamond knives are very, very expensive and they're also very fragile. And this is an image here of our nice ribbon coming off the diamond knife. So we've infiltrated our tissue in advance of embedding with heavy metals. That's called en bloc staining. But after we've sectioned them, the sections are still nearly transparent to the electron beam. So we're going to have to do some positive staining on grid. So on grid staining. This, we use 2% uranyl acetate, followed by a 0.3% lead citrate. We use UA for 20 to 30 minutes, wash thoroughly, dry the grid, 4 minutes on the lead citrate with cooled boiled water, wash it with cooled boiled water, and then dry thoroughly. There's some concerns to be aware of when you're staining on grid. Lead precipitates very easily upon contact with carbon dioxide. So you want to prepare your lead staining chamber with sodium hydroxide pellets to keep the atmosphere dry. Also, we want to avoid lint contaminants on your grid. And we want to do that by using a number 50 grade Whatman filter paper to blot dry. So routine morphology sample prep is not hard, but it is unforgiving because everything that you're doing is depending upon what you've already done and you can't go backwards. So for example, if you fix slowly, you will result in poor membrane preservation and your samples will be unusable. If you have incomplete dehydration, you'll end up with holes in your final block, which may make it very difficult to section, and those holes may enlarge and break the section when you get onto the microscope. And so those sections would be unusable. If you have an incomplete resin exchange and there's solvents left in the block after it's polymerized, then the sections will disintegrate when they hit the water when you're sectioning and be unusable. Improperly formulated plastic can result in soft or gummy blocks, and you cannot cut soft blocks thinly, so that preparation would be unusable. If you're not careful or if you damage your knife, then you'll end up with scratched sections or just unsectionable sections that are unusable. And staining is can produce precipitations of UA or lead. So if you have lead precipitate all over your sections, it's going to look like fine-grained pepper all over your sample. It's going to be unsightly. It's not going to be usable for analysis. So all of these problems have to be solved and mastered. And that's why some people would think routine morphology is hard. But if you're careful, it can be done. The other thing you want to concern yourself when you're doing routine morphology is planning your experiment carefully. And defining your question is the first step. Defining the question, I often get the answer to that question is, I want to see what it looks like. But an electron micrograph of a cross-section cell can be very, very complicated. And so saying, what does it look like, doesn't really get you very well along in analyzing your micrographs. You have to ask a question such as, what does the mitochondria look like? What does the nuclear envelope look like? What does the plasma membrane look like? So be specific when you're defining your question. You want to plan carefully too. You want to make sure that you've got controls so that you can compare and contrast. The other common mistake people make is that I'm going to be able to look at my sample directly. So I'm going to be able to understand what's going on. And that, again, is just not the case because of biodiversity from cell to cell. Things can look be very misleading. And also, you're remembering you're cutting a section of a cell. So on one section of a cell, you may have 100 different sections of different cells in there. When you add it all up together, it's really just representing one full cell. So you want to plan carefully that what you're looking at can be compared and contrasted to things that you know what look like. You also don't want to look for a needle in a haystack. If you are transfecting proteins or mutating genes within your cell population, you want to make sure that you are affecting at least 75 to 80% of the cells in question so that you can find what it is you're looking for. If you've only transfected 10%, there's just no way to guarantee you're going to be able to find it. If you have a situation which does come up when you are looking for a needle in a haystack, then you just need to make sure that it's a really big needle. If you're looking for bacteria within cell, bacteria are quite big and you should be able to find bacteria in cell through a low magnification scan. So you have to plan carefully so that you have a reasonable chance of finding what it is that you're looking for. All right, so let's take a second to introduce a new piece of equipment that CNS has just acquired, the Leica Arctos 3D. It's a new ultramicrotome, which is not that new, but it allows us to service more users because now we have 2 microscopes. One person could be training while someone else can be moving ahead with their work. We will have a cryo stage on this microtome as well. So it's going to be fully equivalent to the microtome we currently have, just a newer model. The Arctos model is also a superiorly engineered microscope or microtome, so it's very, very stable, which is going to allow us to do almost automated serial sectioning, which allows us to do array tomography. Array tomography, you're going to be able to see you'll be able to do collect large samples of serial sections, do 3D reconstructions of cells and organelles, and complex tissues like brain. So when you're going to be doing serial sectioning, you need to have thousands and thousands and thousands of consecutive sections that you can pick up an image without losing one, and also keeping them in order. And this microtome is going to allow us to do it because it is a very stable construction, so it allows for that ribbon to be able to be cut sequentially forever. It also is, we bought this with a special knife, which allows us to insert a slide into the boat that we can then tease these sections, these ribbons onto that slide, many of them, without losing any of them. And then we can take this to the SEM for serial imaging. All right, and again, we can ask 3-dimensional questions now, where before we were only getting 2-dimensional. So another methodology of sample preparation that biologists use is cryo-electron microscopy, and it's often referred to as the Tokiyasu method. So cryo-immuno-EM is going to allow you to label specific proteins within your cell. The things we need to be concerned about here is fixation. So first, as we do with all bio-EM, we have to fix the material. But we can't fix it as strongly as we would for routine morphology, because then we would kill all of our antigenicity. We are going to freeze this sample so that we have a hard block that we can section. So before we freeze it, we want to cryoprotect. Cryoprotection of our material, we're infusing our material with PVP/sucrose. This is a 70% sucrose solution with polyvinyl pyrrolidine added to it. This cryoprotection infuses the tissue, separating the water molecules a bit so that when we freeze in liquid nitrogen, we don't need to go to heroic methods of freezing this. We can just plunge freeze it by hand, and we won't get any ice crystallizations. We'll get very close to vitreous ice if we use a cryoprotectant. We mount this specimen into a frozen microtome, cut sections, pick up the sections with a drop of sucrose, place them on a grid, and then we can carry through with our immunolabeling. Now, cryo-immuno-EM is not cryo-electron microscopy. The cryo part refers to the sectioning stage of this preparation technique. Once we pick up the section, it's thawed. It's at room temperature. But it is hydrated, which is where we're getting the advantage to be able to immunolabel. So we wanted to do overnight on our antibody, wash thoroughly, couple of hours on our secondary antibody, which is a 5 to 10 nanometer particle of gold. Wash that thoroughly, stain that, wash it again. And then this part, the embedding in methicellulose is what makes this basically the Tokiyasu method. We've are now embedding this material in a monolayer of methicellulose. This allows us to air dry the sample that we've cut without causing any air drying artifacts, and allows us to go into the high vacuum of the microscope. This seems simple enough, but to be in reality, publication quality results may take 4 to 6 months, because every step of the way, from fixation to determining your proper primary antibodies, needs to be determined one step at a time, and that just takes time to do. But then you can end up with very high resolution images of your protein localization. So here's an example when you might decide to go this way. You're working in confocal with polyclonal, monoclonal antibodies. You're characterizing that. You've determined that your protein is located to the structure that's surrounding the nucleus. It looks like it's a Golgi labeling, but is it the full Golgi? Is there something more we can learn about this? If we take the same antibody and do EM with it, we can see that we've gained a lot more information that no, this is not a Golgi label. This protein is located just to the trans side of a Golgi stack. So that level of resolution is what you're going for when you're doing cryo immuno-EM. Another example, classic confocal picture of endoplasmic reticulum. We see these lacy distribution of GFP or RFP. But is this the whole story? Is this RFP located just to the ER? You can see we can get 3 different sorts of label or examples of how this, what could be labeled in this image. In this picture here, we see the ER nicely, but it's just the cargo within the ER that is actually labeled. That may look exactly like this picture. In this picture here, we see that the large lumen of the lamellular ER is not labeled as much as these circular budding portions of the ER are labeled. And that might look exactly like this as well. Finally, in this last picture, we see that the lamellular ER stacks are heavily labeled, but if you look closely, it's actually the luminal side of the membrane that is being labeled. So all 3 of these different labeling results could ultimately have looked like this by confocal. So that's another reason that you can consider doing immuno-EM. So the conditions that have to be met, you need to determine the level of fixation that your can the antigenicity of your target molecule be preserved by fixation? Is there enough antigen present in a 50 nanometer thin section? So what looks like heavy label in a whole cell may actually not be enough when it comes to a 50 nanometer thin section. If you're making antibodies or buying them, are they clean, specific, and concentrated enough? These are conditions that have to be tested. Will GFP work? GFP, there's a lot of really good anti-GFP, anti-RFPs, anti-HET, anti-MIC antibodies that are working at the EM level. So if you've got a clean, clear localization of your GFP, going to EM is probably going to be successful. You also want to consider what your positive and negative controls are. A negative control is going to generally be a sample that's labeled with secondary but no primary. Positive control can be a little bit different, something that you know that works under these conditions, under other circumstances. What your positive control is helping you with is troubleshooting when things go south. If you have a sample, everything should work, but it doesn't, but your positive control did work, well, then you've got more information. If your positive control didn't work, now you have information too that you can use to design your next experiment. Lastly, do you have good sampling? Again, if you're doing transfected cells, you want to make sure that you've got 75 to 85% of the cells are expressing that transfection. So this would be a typical way that you might present in publication your immuno-EM data. You would have your routine morphology that gives you a high resolution, high contrast image of your cells. You would have next to that your negative control that shows that there is no labeling. And then finally your test sample. If you're using very small particles of gold, pointing that out with arrows is always helpful. So at CNS, we have electron microscopy training and services. The routine morphology studies, as I said, would take about a 2-week turnaround time. Negative staining studies are less than an hour turnaround time. Immunogold would be 1 to 6 months turnaround time. It involves a lot more interactions with the user. All users can be trained to do this work either completely themselves or in partnership with myself. Negative staining, users can be trained to do that very quickly. Routine morphology, if you're just doing one experiment for a quick look at something, it may not be worth your time to invest learning how to section and embed the material. So I could do the embedding and sectioning, and then you could be trained on the microscope. Or you could be trained to do the whole thing. Immunogold, again, is a large dedication of time, and you'd have to weigh that how you would want to go about getting that done. So I thought I'd change now to scanning electron microscopy. Scanning electron microscopy is not heavily used by the biology world so much anymore. The classic example is Drosophila eyes. Drosophila eyes are susceptible to genetic mutations, particularly in oncogenes, and those mutations are expressed beautifully in Drosophila eyes. Either these little spikes can be damaged, surfaces of the eye itself can be damaged. And this is what most classical scanning EM would be like. Sample preparation is very simple. It can be done in less than a day. As always, we start with fixation, then full dehydration to 100% ethanol. We need to load our sample into some sort of drying chamber that will fit into a critical point dryer. We critical point dry the sample, remove it, and mount it on the stub. Mounting it on the stub may be really the most difficult part, because once your samples are critical point dried, they're extremely fragile. And biomaterial has a lot of moving parts. You've got, if you're looking at a fly or something along these lines, they have antennae and little legs and little hairs. And you don't want to damage that when you're mounting it on the stub. So this part takes a little bit of practice to get used to. But then you sputter coat the sample and you put it in the scope and you've got your imaging. So while SEM is not highly used by biologists, I would encourage you to think about adding it to your studies. This is a study I was working on with Mary Gehring at Whitehead Institute. She's studying Arabidopsis and the epigenetics of seed formation. And we decided to do some SEM on her plants. So there are 2 ways a seed can be produced within a flower. It can be the flower can be cross-pollinated, or it can self-pollinate. In Arabidopsis lyrata, we can see from this picture that this plant can never self-pollinate. This structure here is the is the pistil where that receives the pollen. These structures here are the stigma that produce the pollen, or the anthers that produce the pollen. But while the stigma is ready to receive pollen, the anthers are not producing any. So this could never self-pollinate. In the Arabidopsis thetalinea, the pistil stigma is ready for accepting pollen, and the anthers are producing pollen. The pollen is the little purple dots. So this plant can self-pollinate. Now, this information can be gained through epigenetics and through molecular studies, but adding a picture like this to your final publication can really set it apart from your competition. There are plenty more sorts of sample preparation methodologies that can be used for biologists, although I refer to them sort of as odd ducks, but they're very, very specific applications and are not generally used that much anymore. Freeze fracture, for one, where you freeze your cell, hit it with a knife, crack it open along the planes of least resistance, typically the inside of the membranes. This work was classically done to show nuclear pores, seen here. We also can make platinum replicas where we place the sample onto a cover slip, coat it with heavy metal, platinum, dissolve away the cover slip and all of the real biological material, and then pick up the replica that we've made on a grid to look at it on the TEM. Produces very high contrast, high resolution images, is not easy to master. You can also use this technique to look at the cytoskeletal structures of culture cells. Another methodology that's gaining popularity is called CLEM, or correlative light electron microscopy, where you're looking at the exact same cell by light and then following it by electron microscopy. This technique has gained so much popularity that the microscope companies are actually developing instruments to make this procedure easier. None of these procedures are particularly easy and are basically done by people who that is all that they do. They've mastered the technique and that's what they've stuck with. So, in conclusion, the most difficult aspects of bio-EM is the sample preparation of the specimen material. So once a number of basic techniques have been mastered, the beginner can easily be trained to use the electron microscope. You want to define your questions clearly. You want to choose the correct preparation techniques and determine the controls that you use to aid you in analysis. But keep in mind, there are very, very few biological research problems that do not benefit from the use of electron microscopy. If you are interested in including EM analysis in your research project, I encourage you to please contact me for a consultation. We can communicate with email now while we're all separated, and in person when once we can get the lab back open. If you try and include any supporting information, papers or images, then that will help me understand what your imaging goal is. And lastly, I'd like to do a shout out to Sandra Castone. She's going to be giving the seminar next week on Introduction to 3D Printing with Formlabs. So we look forward to learning about that. All right, so then I'd just like to say thank you for your attention, and I hope that I can be of service in the near future. Thank you.